So today we'll be talking about cell cycle regulation and how it's connected to cancer cells. The timing and rate of cell division is crucial to normal growth, development, and maintenance of the cells. Many cells divide differently and more often based on what they're used for. For example, skin cells frequently divide, while liver cells go faster if there is a wound but don't normally divide as often. Some specialized cells, like nerve and muscle cells, don't divide at all. Understanding cellular regulation is what gets us closer to understanding cancer cells. The events of the cell cycle are directed by cell cycle control system. This is kind of like an automatic washing machine. The timing device on the washing machine is like how the cell cycle continues on its own with an internal clock. The sensor on the washing machine when there's too much water is like the external adjustments that the cell cycle makes. Within the cell cycle, there are many checkpoints, and the checkpoints are where the stop and go ahead signals regulate the cycle. Animal cells have built-in stop signals that stop the cell cycle until they are overridden by go signals. There are three major checkpoints, which are the G1, G2, and M checkpoints. The G1 is the most important checkpoint and is considered the restriction point in mammals. If the cell receives the go signal, it will go through the G1, S, G2, and M phase and continue to divide. If it receives the stop signal, it will go to the G0 phase, which is a non-dividing state, and most cells are actually in that phase most of the time. The G2 and the M phases just carry out the rest of the division process. PDGF is a growth protein that is released so other cells will divide. It is made by blood cell fragments called platelets. This is required for the division of fibroblasts in culture. This also allows cells to pass the G1 phase and divide. MPF stands for maturation promoting factor, and it triggers the cell's passage past the G2 checkpoint into the M phase. Another two molecules that control the cell cycle are protein kinases and cyclines. Protein kinases are enzymes that activate or inactivate other proteins by phosphorylating them. Some of these proteins give the go-ahead signals at the G1 and G2 checkpoints. To be active, a protein kinases must be attached to a cycline, which is a protein that gets its name from its cyclically fluctuating concentration in the cell. These kinases are called cycline-dependent kinases. MPF is also the cycline CDK complex. All of these things come together to control the cells, but cancer cells do not listen to the normal signs that regulate the cell cycle. These cells divide excessively and invade other tissues, which is why the organism in it gets sick or dies. Cancer cells also don't stop dividing when growth factors are depleted, like a normal cell would. Scientists hypothesize that either cancer cells make a required growth factor themselves or have an abnormal cell cycle control system. If cancer cells are given a continual supply of nutrients, they divide indefinitely and are essentially immortal. The cancer cells result from an interruption in the normal cell cycle. The body is not meant to have cells divide indefinitely, which is why people get sick. So that is an overview on cellular regulation, and I hope that helped.